So I want to start off with that genetics and study of genetics has helped people in so many ways. It has led to artificial insulin, which is how almost all the insulin produced, which has saved tens of millions of lives, is made currently. It has led to new HIV treatments, which have saved millions of lives. It has led to new cancer therapies. It has way reduced the incidence of Tay-Sachs and a couple other horrible diseases that kill people before they're 5 years old in painful, debilitating ways that there is no medicine for. However, there's a dark side, especially with the marketing and the fear factor that people promote, where they take advantage of people and may even promote eventually eugenetics with genetic research. And I want to highlight one specific example with Spark and autism, and how genetic research in autism has really hurt the autistic community. First, and I'm going to take this by a scientific approach. I'm not going to talk about too much about the social thing, just the science and how this is a bunch of pretty much pseudoscience. So let us start with talking about the science, and I promise to make this as understandable as possible, even if you have no background in science. Well, first of all, how do we analyze genes and genetics? It's not just, as many people paint it as, oh, something looks different, this gene must be important, or something. Difference is everyone's genome is vastly different, and everyone has mutations. But what is meaningful and how do we analyze it? Well, let me give you an example of the confusion we get. The word hi. Let's pretend we know the language. Hi. If I mutate into the word hello, big difference in how it looks, but not a big difference in what I've expressed. If I take hi and I say it twice, hi, hi, or I can say it even 40 times, hi, 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 still same thing. A small change, I add a T at the end, from hi to hit, huge change in what they expressed. I can also just sometimes repeating something will make a big change. Like if I say hit it, I repeat the IT, that means something different. I could say hit. I could say hi, hi, hi. The it's H's that I might be make it bigger. Or hi, hi, I'm starting to express something different even though I repeated something. Hello, if I added space in between, totally different thing. Then I have another example. Hi, Gad, my name. Add a T in the middle, hit Gad. Delete an H, IGAD. IGAD is more similar to hi, Gad, I would say. Still big difference, but deleting made a big difference in that case. But in that case, if I add a bunch of random letters, LLL before hi, Gad, it makes almost no difference. So having a big mutation might make almost no difference. A small mutation might make a big difference. And how do you line up the genes? Like, if I see this LLH GAD, do I start off writing L under it, and I see that it doesn't match up nicely? Or do I match it up in some fancy algorithm? Well, we really don't know a good algorithm to do this just yet. We're getting better at it. But a good algorithm would mean that we understand the genetic code, and we don't. The way we usually do this around is we need a large sample size with just one type of mutation. So you can really example one mutation versus not 1,000 people with the same thing, but each of them have a different mutation. I'm saying 1,000 people with the same thing and the same mutation or similar. Or we have to do experiments in mice or flies or more organisms that we can really compare to human beings. And with autism, there's many different genes that we think could cause it, so that's simply not doable, even if we have this ridiculously sized population. A lot of the ones we know are from less than 100 people, and this doesn't work because we don't have a good animal model of autism. How do you know autism has to do with speech, language, communication? It has to do with social sensory things that we just don't see the same thing in mice or animals. And how do you quantify those ones? So we really can't do these ones, the same good genetic analysis. Also, a lot of traits, how they exactly manifest depends on many genes, not just that one little gene. And I'll get how this manifestation is a big deal later on. There's only one gene that can give you blonde hair color. One gene gives you blonde hair. There's 2 possible genes that could do it, but you only need one of them to get blonde hair. All other genes just control the degree of blonde hair you have. The same as with red hair, the same as with brunette, or the exact size or how it's expressed. There's often almost every trait is controlled by multiple genes, and that's really what autism has been pointed out, that it's controlled by many genes. Not a single gene makes you autistic in most cases. So that makes the whole analysis here even more complicated and hard to tell. So fine, the genetics is hard and how it's going to expressed. Well, even more, we have something called epigenetics. So epigenetics is the study of how much a gene is turned on or off or expressed in different ways. So genes make proteins and proteins make you. There's in between with something called RNA. So genes make RNA, which makes proteins, which makes you. Proteins make your skin and everything. How much a gene can be turned 0% off, 0% on, nothing's made, 100% on, it's made at full speed. It can be turned on in between amount, 50, 60%, 70%. It can be turned on so it makes a different protein or expressed differently. There's different ways the same gene can be read. For example, I can say hi, but maybe I say hi something else afterwards, which changes the whole meaning of hi. That same hi, but it's just read differently in context. Context in language is important, and so is in genetic code. Well, epigenetics is that study of just how much a gene is expressed. There's many ways that genes can be expressed, and I'm just giving you a broad overview. These are not meant to be the most accurate pictures, but they're meant to give you an idea and how complicated it is. DNA is coiled. But DNA can be coiled in many different ways. This DNA here, it can be coiled so that it's either more coiled or less coiled. If it's less coiled, oftentimes that turns on a region of DNA more. The way that this turns on a region of DNA more is that it's easier for the machinery that reads the DNA to actually read it. It's the same thing as if I have a book and I increase in the font size is really small, tiny, and everything, all the words are compact together, it can be really hard to read. If I make a bigger font size and everything is more spread out, it can be a lot easier to read because I can see it better. The same as with the machinery, you can access the whole thing, you can see it better, it can move down the track quicker. When it doesn't have to turn, it can just go straight down the track. Imagine like a bump, machinery like reading on a flat railway track instead of like a bumpy road. Or I can do the opposite, where I coil it even more. When I coil it even more, it becomes less accessible, maybe even inaccessible for stuff to read. I can not only coil the DNA in a normal coil, I can have bigger coils where I take the DNA strands and I make either big loops. And these loops can expose more surface area to make stuff more easily. Or they can loop around itself and make it less accessible, only make it read certain portions of the DNA. And then I have all this, I have something called histones, which are proteins that this DNA can wrap around and expose different parts of it. So that's one part of epigenetics, turn on of genes. We also have axonal modifiers of DNA on that, and these are chemical modifiers, such as we can add a methyl group, CH3, we can add an acetyl group, which I show here. We can add other groups, and this can make the things that read DNA either be more attracted or less attracted, or it can make it bound more tightly to histones. It can change the packaging. It can do all these different things. And we don't fully understand it, but it's clear it has an effect. Other way DNA can be modified is DNA can have lots of things bound to the DNA. And these lots of things, when they bound to DNA, they can bring in stuff to read it more or read it less. They can expose regions more or less. or they can just attract chemical things again. RNA splicers, so DNA makes RNA, which makes proteins. The RNA can be cut up and put back together in different ways, and these can make the same code be read differently. Imagine if I take a book and I just randomly start taking away letters or words. I can get a very different sentence or different meaning out of it. I could change, if I drop the word not, I can totally make this thing the complete opposite. That's what RNA splices have the ability to do. And some of that is inherited, some of that is interacting with other things. There's things that can stabilize RNA. So DNA makes RNA, which makes protein, but an RNA might only make one protein, or it might be stabilized so it can make 100 times the amount of protein. It might be stored for later use. We have signs that the body stores RNA for later use. Or there's other chemical modifiers of RNA. There's even proteins that can be partially inherited. And a cool thing and complicated thing about all of this is that it can be partially inherited, and it partially can be from the environment. We do know that even though this is complicated and we do not know how it works in the human body, we do know it's a real effect because we've done it in simple things with mice, flies, bacteria, plants, everything, in very controlled environments. We also know in extreme cases like Prader-Willis and Angelman syndrome that epigenetics plays a huge role. Same gene mutation, if it comes from expressed in one way, it leads to totally different symptoms than if it's expressed the other way. One way it leads to a huge weight gain, and one way usually leads to a huge problem with anemia and malnutrition and not eating enough. Another way, you eat too much, oftentimes. So then we also have its symptoms in, like we have one example in mice where it's even more complicated, where it depends if you inherited it from a mom or dad. If you inherited from the mom and you had a poor diet, it leads to the offspring being overweight and unhealthy. If you inherit it from the dad, it's the opposite. The same gene, just that the epigenetics is inherited different from the mom or dad. And this was in the Guti mice where we repeated the same gene multiple times and did some other things to make it more extreme. So yeah, it even depends on who you inherit the gene mutation from. It's real, and we also know it's real because epigenetics literally what tells a nerve cell that it's not a skin cell, that a skin cell is not an intestine cell. It's what tells every cell what it is and helps in development. But we don't know how inheritance, because you can inherit some of it from your parents. We don't know how that interacts with environment, because some of it is how your environment, what you do in your environment impacts epigenetics. And we know it with extreme example, like in mice, if the parents are fed a lot or fed little, it impacts how their offspring are through epigenetic means, and you can see it a couple of generations later. That's well established, but these are the super controlled mice, and we know exercise has an impact. But how does exercise plus diet work together? It also varies on your genetic makeup how big this epigenetic is, and it's so much more complicated than something like a human being. Then we also have in epigenetics something called stochastism or randomness. Same thing, stochastic is the fancy word for random. And that is that some of these epigenetic markers are not simply inherited or not inherited, or done by your environment or not done by your environment that your parents grew up with. And your parents' environment doesn't affect how you grow up. It is done by just randomly some genes are left with epigenetic markers on them, and some of them are randomly erased. If something is weakly bound, it might be erased, it might stay on. Same idea, a flip of a coin, whether inherited or not. And that can lead to markedly different outcomes in how something develops. And we see this a little bit with stem cells, but it also could be something that happens even not in inheritance, just when you're born, early on in development, some genes will randomly turn on and off. All this could happen, but this means that a single system with the same genes could end up being radically different in different individuals, and we're not taking account environmental impacts in the room or how a child is raised or anything. Just this alone means that the same gene could radically manifest differently, which means it's even harder to figure out, is this gene causing autism? And what type and how does it impact it in a more individual level? But that leads to a huge fear. Let's say I have a child and I want to figure out that this child, I want to diagnose not only that they have autism, which I've always suspect that these tests cannot do very well, and I'll even give more reason, but I want to say how the autism specifically manifests. Do they have do they have more communication problems or more sensory overload problems? Well, I can't, because if I have a small sample size of like 5 or 10 individuals, all of these things, the multiple genes interactions, the epigenetics, the environment they grew up with, could all totally throw away my data. But I might find a pattern. It's very easy to find a pattern. A lot of these individuals, you only have like 10 or 100 individuals. Sometimes there's even, a lot of times in these spark studies, there's single digits of number of individuals with this mutation. And what you see is, oh yeah, I know that all these individuals have something, so it must be caused by this gene mutation. That is a horrible way of doing it. If you give me any random group of 4 people, I can find a bunch of things, and I challenge you to do this, how they're similar. And then I conclude that it's because they all have one thing. If I have 4 individuals with glasses, for example, I can find 10 million things they probably have similar, and then conclude the glasses cause you to all be male, all be female, have a certain gender identity, have a certain sexual identity. I can make up all those crazy things, but they're false, and we can really overinterpret the results here. But that leads into fear factors, that leads into people not getting to know the actual child, not helping it, but it's a very profitable way of looking at it, because then you don't have to actually get to know the child or diagnose them. And then we look at the actual data set. Well, the data set is highly biased, which leads to even more problem within analysis. So autism used to be mainly diagnosed in male children. Until lately, some people thought autism was only in men. And when it first was discovered, people thought it was only in men, in male children. But now that we went from like a ratio of like 50-1 to like 4:1 to 3:1 to now some people say 2:1 ratio of male to female. And I know they say male to female to report the numbers. I know gender is a whole thing, and it's more complicated than that. And there's also transsexual individuals, and it's not a nice way, but I'm just saying how the numbers are reported. So the samples are biased, which means when you put in biased samples into not a good analyzing method, you're going to get biased results, and you're going to get these tests might not recognize autism in certain individuals. It might not recognize it in females, or might not recognize it in transsexual people, or might misdiagnose them with a specific type of autism, which is also very questionable, or if they mask it or hide it. Also, autism has been mostly diagnosed in white Western children, and bias samples have been a huge problem in genetics for so long. For example, we used to think there was only one gene that caused you to be blonde or not. Now there's 2 genes, but that's because we only looked at Western blonde individuals from Europe. We found on the Solomon Island there was a different genetic mutation in a different gene that could also make you blonde. Totally independent. Never knew it, but we thought for so long that they just had the same blonde gene. One individual a while ago pushed down the common male ancestor of all guys to over 10,000 years before we thought it was. One guy somewhere in the south, I forget exactly where in the south, of African ancestry, because most of the people that have been sequenced thoroughly are from European ancestry. Yeah, so we're going to be missing a huge amount of data, which means that many individuals with autism are going to be overlooked And the more we rely on it, the more we're going to overlook those individuals. And the more we're going to overanalyze specific traits that autistics might have with gene mutations that do not necessarily cause them at all. Secondly, the sample size is very small, which is always a problem. Very small. Not only is it biased, oftentimes there's only like 10 or fewer individuals with the specific mutations. The people that are in the samples are usually it's the parents that push them to get this test, or parents are pushing for it, which means that people that might have a different, which might lead to bias in the forms. It might be the parents that can deal with it less are ones pushing for it. So it might be kids with forms that need more medical attention or less. And oftentimes they're unconsenting. Most of the individuals in this study, almost all of them participants, and I'll show it later on, like easily over 70% of them, are children that it was the parents that consented to it, not them. So that means that, again, we're not looking at people that necessarily want it, and the data about how it manifests and stuff is totally different than what the children. Children, when they grow up, oftentimes report very different experiences than what the parents say. And oftentimes, the parents talk about how awful the autism was when the kids say it was the society's fault. And that just leads to a lot of bias. And also, should you ever have a sample that is unconsenting, on the actual website, there's information about how to collect spit from unconsenting children, or children that are not willing. They don't say unconsenting, but children that are not willing to participate in the study. And they also talk about getting permissions for when you have legal guardianship over an individual after the 18, if you still have certain rights. So even more than the under 18 report here are getting this. So huge problem with the sample, huge ethical dilemma. It makes it even more biased. And then they're going to exaggerate it out of profit. And I can show you already that profit is a huge motive of this, and they're not speaking for the autistic community. So let's look at let's look at the board of directors, meet the staff. None of these staff members are autistic. None of them. They all work in the study of autism, and there's no one adult. And how do you have one of the biggest autistic projects has no one on your board, on your staff, that you can list that are autistic? Instead, you have multiple people related to they're making huge money off of genomic studies and control scientific journals, get money off of diagnosing, and all these other things. Have given TED Talks. They really have made a career of making money without hiring any, which is really strange. Like, if you make a career of any individual, you should know some people of that group and be able to bring them on board. Very, very weird. Look at some of the advisory boards. Same deal. Is a relationship codes, National Human Genome, but again, not autistic. The closest is a parent of a 17-year-old daughter with this syndrome. Then she explains the diagnosis. And then she wants drug treatment and clinical trial designs, and many autistics do not want this at all. They do not want to be treated with drugs. Really, really weird. Then you look at the research partners, you find even more messed up things. When you look at the research partners, you find that Autism Speaks is a huge one of the research partners. Autism Speaks, let's look at the board for a second. None of these people on the board are autistic. This is from their own website. However, you have the AMC network, and AMC as the AMC that does movie stuff. You have Mobile capital management, yeah, the private investment firm, you have Goldman Sachs somewhere, you have another realty estate, you have Ventures. All of these people are multimillionaires, and people on TV saw almost none of them, Goldman Sachs, here it is, almost none of them have any medical degrees, and they're funding this, and they make a lot of profit. And if you look at the funding from Autism Speak website, look at the funding, over 90% of them goes to advertising and paying themselves. Less than 1% of it goes to helping, giving accessibility resources to autistic families, which a lot of autistic people have put to doubt, because one of those resources, for example, is if you have an autistic child, this is how you go for grief, and treat it like a death. And it says stuff as, don't blame the autism. I'm sorry, don't blame your child, blame the autism. Legit word for word in the statement. Yeah, these seem like biased part. Autism side. Oh yeah, Autism Speak. One of the spokespersons' husband has been known to sexually harass at events autistic people, and has been caught multiple times on messaging boards. Autism Society, same deal. Autism Science Foundation, same deal. None of these societies are led by any autistic group at all. None of them have any autistic people on the board or anywhere give support for basic resources. They're all about stigmatizing it. Yeah, and then we look at the participants. Look, they have 90,000 individuals with ASD. Okay, 77,000 of them, it's the parents that gave them consent. We know of because of the AIDS. Very few of them are actually directly related, where we can do a very good summary, but that's not going to tell us very good details. And that's still, we don't have a big enough population of non-autistics to really compare them to, because you also need a huge population of non-autistics to see that it's not just a fluke in the data. I really suspect it. And then I look at some of these reports, and I've seen parents say my kid has the worst mutation ever in MTHFR, and I'm looking at it, and it makes it the worst one, and they're homozygous for it. And I'm like, that means they have 2 mutations of this gene. And that mutation, I know, from the data they publish, is 25% of Hispanics have that mutation, and 15% of Caucasians. It can't be that much of an effect if that's going on. But they fearmonger with these mutations because that gives them profit and gives them stuff. Instead, what this money is, and they're funded tens of millions of dollars every year, that money should go instead to better diagnosis, the traditional method. Just traditional diagnosis, which is a lot less likely that we make up a genetic test that misses a bunch of individuals that don't have the same gene mutation that still have autism. Or that gives us false positives even, because we just used a biased sample. It would be much better if we went to accessibility services, or to the autistic community in general, or to health sources, or the money went to actual good research for cancer, something like that. This is about fear-mongering to people, to profit off of people based on not real data. And that was just a brief start of all the genes. I think you could see there's many genes, and too many genes just to find any strong correlation. So yeah, this is just not going to help anyone. And I hope you understand why I'm against this study, and that it's done with many, many non-consenting individuals. Thank you for listening, and please really hit the subscribe button. It really helps me out. And like and leave a comment behind. Thank you very much. And I will link the sources below.